I'm excited for a great discussion today. Thank you for joining us.
Awesome. Thanks, Jen. Shamal, I can start off if you want. I think first of all, I want to just say a big thank you and big kudos to Jen, Michelle, everybody at the Dime Society. You guys are rock stars, and we really appreciate everything you guys are doing. You have a big fan club over at Pfizer. And I want to say thank you to everybody joining. I hope everybody's staying safe. A little intro about myself. My name is Nikhil Mahadevan. I'm a data scientist over at the Digital Medicine and Translational Imaging Group at Pfizer, located in Cambridge, Massachusetts. My background is focused kind of in software engineering. I started off building mobile applications and then transitioning into cloud applications, now intersecting with data science. And to end it off, I guess I'm definitely very motivated to make use of digital devices to monitor patients outside of the clinic, and looking forward to a great discussion today.
I just want to second what Nikhil said about the Dime Society and this sort of platform that you guys have created for us to share ideas and have discussions like these. My name is Shamal Patel. I lead the data science team in the Digital Medicine and Translational Imaging Group at Pfizer. And I've worked with Nikhil and others on essentially taking digital endpoints all the way from idea to deployment. So that's sort of the spectrum that we cover. And this specific work was focused on our work in Parkinson's disease. This was early on in 2017 when we both joined this group at Pfizer. And then since then we have expanded into a lot of other areas, and so we're excited to share sort of this early work that we did, and a lot of the lessons learned from this work have been very helpful for us, and hopefully we can share some of them with you in terms of how to bring digital endpoints into clinical trials. So super excited to share this and have a great discussion with you all. So
awesome. Shamal, so it looks like I'm driving. I will just tell me if you guys want to kick it off. And,
let's go in. Yeah. So first, I just want to take a moment to acknowledge all the people who've contributed, colleagues, collaborators. So our co-authors who had very direct contribution in terms of writing, brainstorming, and coming up with some of the ideas that we have presented here, and also the larger Blue Sky team. So Blue Sky was a project that was focused on Parkinson's. Was the first project actually that the Digital Medicine and Translational Imaging Group kind of championed at Pfizer. So Peter Bergathon, Dan Carlin, some of these folks are not at Pfizer anymore, but they had a significant impact on this work. So just want to make sure to take a moment to acknowledge them. Can you move to the next slide? All right, so just a quick overview, and again, I'm not going to talk about Parkinson's. A lot of you might already know more about the disease and manifestations, but Parkinson's is a very interesting condition for digital endpoints because the range of symptoms that are present in PD are so broad and so diverse from patient to patient. So it is very hard for you to use one tool to measure or measure just one aspect of the condition and get a full picture of the patient experience. There's a lot of work that has been done in this space, and I think that's one of the reasons why PD is a great candidate for one of the first conditions where we would want to venture for digital endpoints, because I think the first paper that I've read in this space for PD was published in 1980, where they were trying to use accelerometers, very early versions of these accelerometers, which are kind of big and bulky for measuring tremor, quantifying tremor. So there's a lot of groundwork that has been done by researchers in academia and industry in terms of trying to use technology to understand various symptoms. So that's, that gives us a good foundation, and we definitely used a lot of the foundation for this work. The main challenge that we encountered when we started off on this path was one of the, despite all this work, they're still lacking in a few areas. One was sort of, how do you, go from, a lot of the work has been done in the lab or the clinic setting to validate methods during the performance of prescribed activities and prescribed tasks, even ADLs that were prescribed. And I don't think those really reflect well, what happens during daily life. So in terms of translating this work from lab to the home, there is a gap, and there are recent works that have started to close the gap, but we identified that as one of the challenges why we had to go and do something on our own in this space. And the other aspect was the use of sensing modalities. So the use case we were considering for our work was clinical trials, and how do we measure, how do we derive digital measures of motor symptoms in a clinical trial, ostensibly at a large clinical trial where we would have to deploy this technology in several hundred patients across a large geographical area. So there were other constraints in terms of, we would want to keep the system fairly simple, and not try to measure every aspect of the condition. And a lot of the work that has been done in this space in the past has been, not really thinking about, okay, how do we constrain this to think something that is simple and scalable. It's always driven by, okay, what can we measure, and how accurately can we characterize all different aspects of the condition. So the approach that we took, with this in mind, was one, because we wanted to measure motor symptoms during daily life. We wanted to have a good, that we wanted to use context to drive the assessment of symptoms and not just measure symptoms continuously, because I think context, one of the challenges in daily lives is actually the bigger challenge in deploying digital endpoints is knowing when not to measure. Continuous measurement is sounds great, but I think in daily life there are lots of situations where you are like, okay, you definitely don't want to measure symptoms under these conditions because those measurements are not really valid. So we wanted context to drive assessment of symptoms. That was one. We wanted to keep the architecture fairly modular. So we wanted to tackle multiple symptoms, but we also wanted to develop them in parallel and then figure out how to put them together into a single solution that would be used for measuring digital endpoints. So we kept the architecture fairly modular. We could combine pieces, and we also kind of developed these modules somewhat in parallel to each other. And then the third thing was we wanted to keep the technology simple and scalable, so that we can minimize patient burden and make it easy for from a logistics point of view in terms of deploying this in a clinical trial. You want to go to the next slide? Yeah. So this is sort of the overview of our approach. Just a high level overview. We kind of made a choice of relying only on accelerometer data. A number of reasons for that. One of the primary reasons is accelerometers are extremely low power, and that helps, from a monitoring point of view. You can give a device to somebody and they can wear it for, some of the devices can be worn for a few months now without having to recharge them. So just from a burden point of view, it's just, you put the device on and you go. So that was one of the key drivers. We also made sure that, from a measurement point of view, we could measure all those things that we are interested in using accelerometer only. And then we drill down into sort of what is it that from a context point of view, we would need to know. So there are two things that we identified that were critical. So we wanted to make sure we use the presence of hand movement as a way to filter out, because the two symptoms that were of interest to us were resting tremor and bradykinesia. So hand movement was one way to filter out a situation where we would want to discard any periods where the individual is engaged in some activities where their hands are being used for the assessment of tremor. And then the other condition that we identified that would affect the assessment of bradykinesia was even when your hand was moving. There are certain, like there are a lot of scenarios, I think, where you could imagine that you might want to filter out, but one of the key scenarios was gait, and gait is something that you would not use during the assessment of bradykinesia. So we wanted to make sure that if the person was walking, we would exclude those periods and use the rest for the assessment of bradykinesia. So that this is sort of the high level architecture, and there were definitely, you could, I think one of the points of, you could argue, you might want to add additional context to this, and you certainly can, if you have other conditions that you want to, situations that you want to use or exclude. And hopefully sort of the modular architecture helps you if you want to use this framework for your own work. So I'll stop there, and I'll let Nikhil kind of walk you through some of the key results that we achieved.
Yep, thanks. So I can walk you guys through the key results from our work. We'll try to keep it to just the highlight reel. But basically, the first aspect here, and this is actually kind of interesting, and we definitely would be interested in hearing your feedback on this concept. But as Shamal said, there's a lot of work out there that's been done. We have devices out there that have been proven to be able to measure people at home. But one of the aspects that we focus on is how do we say that our measurements are actually meaningful, clinically meaningful and useful. And the way that a lot of people do it right now, and this is the way that we started off with doing it, is comparing it to current assessments of disease severity. And I think it's an interesting kind of idea because we're in a bit of a transition here where, we have the current assessments, which have its own limitations, and we're creating a new paradigm of assessments that are kind of new, but we still need to relate it back to the previous assessments. So yeah, definitely looking to hear some thoughts on that. So specifically on this is, we basically started off by comparing our measurements to the MDS-UPDRS Part 3 scale, which is specifically the motor section. And we looked at 3 scores. We first looked at the resting tremor constancy score. Number 2 was the rest tremor amplitude score, and bradykinesia score. Couple things on this. The constancy and tremor amplitude, all scores are rated on 0 to 4, an ordinal scale. Bradykinesia is measured all across the body, and there are respective scores for each. Since we're specifically focused on a wrist sensor only, we decided to dwindle down to just the scores that are related to the hand. So we ended up comparing the bradykinesia scores to the summation of 3 sub-UPDRS categories, specifically finger tapping, hand movements, and pronation, supination. And overall, we saw, which was comforting to us, we saw pretty good results, pretty good agreement between our measures of, our digital measures of tremor and bradykinesia in comparison to the clinical scores. The second aspect that we looked into was being able to assess whether these measurements can track medication-related changes. And so with Parkinson's disease, we decided to dig deeper into patient-reported states of on and off. On meaning when your motor symptoms are minimized, and off when they're not. And we wanted to basically say, hey, do these measures reflect the changes that you would see in both of these states? So our clinical study basically had subjects come into a lab when they were self-reported on, and then also a second time when they were self-reported off. And we basically ran these measures in both of those states and looked at the differences. I can get into these plots a little bit more. Basically, what we were looking at was the differences between the on and the off state. So for example, with tremor constancy, in the on state, you would hypothesize a lower tremor constancy, and then in the off state, you would hypothesize a higher one. So when you take the difference, you would see a negative number, and then vice versa when you look at off minus on. And so that trend was apparent with the calculations of tremor constancy. The left plot, tremor amplitude, which is plot B. And then plot C also, it was apparent, but to a lesser degree. So that was some interesting notes on that. I think, one of the reasons for that is, as I said earlier, bradykinesia is apparent across the body, and we're specifically only looking at the hand related, so we might be missing some aspects of it. But generally good findings nonetheless. And lastly, I think this is kind of the bottom line here. We're at Pfizer, we're looking to augment the assessments of patients during clinical trials. And right now, the current assessments have limitations specifically, and I think this is a common thread in a lot of the work of the DIME members and what everybody's been showing. But basically, they're subjective. They require a clinician, and this might be more prevalent in CNS-related disorders. They're subjective. They require a physician to assess the patient. They're episodic, so it kind of only gives you a window in time of how the patient is doing, because they have to come into the lab to see the physician. And they're very kind of rudimentary. So like the UPDRS scale, it's on a scale of 0 to 4. And so really what we're trying to do here is do a bit of a paradigm shift in how we measure patients. And so we're switching to this, wearable device that somebody can wear at home. And the main advantages here are it's objective. It's purely from a wearable device. It's continuous. You can strap this on a patient at home for 1 to 2 weeks or however long your use case demands. And 3, it has a higher granularity. So you have the ability to see fluctuations that you wouldn't normally see with the current assessment tools. So just to describe this graph a little bit more, so this is one subject from our study, and these are our output measurements from the system that Shamil described. On the left-hand side was when the patient was self-determined to be in the off state, and on the right-hand side was when the patient was determined to be in the on state. The first row is tremor-related measurements, and the second row is geared towards bradykinesia, specifically highlighting hand movement related measurements. So we do see, what you would hypothesize in terms of the changes. So more higher tremor detection and constancy, as well as amplitude while they were in the off state. And so you can kind of see that reduced in the on state. And with the hand movement related aspect, we see lower amount of hand movement in the off state, which relates to being more bradykinetic, and then in the on state, we see much more hand movement, meaning they're more fluid and able to move their hand freely. So this is just an example of how we thought this type of assessment tool could be used, and was really trying to highlight the aspect of it being objective and continuous versus the current assessment paradigm. Lastly, we just wanted to call out some links that were really helpful to us. So the first one is from Solarian's group, and big kudos to these guys. They did really great work in terms of making their work reproducible, and this served as the foundation for how we built our tremor algorithm. The second one is from Sylvia Deldin's lab, also must read in our opinion in terms of a good review on wearable devices and monitoring Parkinson's disease at home. And lastly, that is our GitHub link to the code we used to create this work. We definitely would love people to download it, try it out, iterate on it, give us feedback. We would love that. And with that, I think that's pretty much the high-level overview. I think, yeah, this is for the end, so I can throw it back to you, Jen, I believe.
Fantastic. This is terrific work, guys, and thank you for taking us through sort of all of the decision making and things that came before the data capture, as well as the findings. And also, it's terrific that all that data is available through GitHub. Thank you for making that resource available. Folks on the line, this is where I'd like to start giving you the opportunity to pose your questions. I've got the chat box open here. And also, if you want to ask a question directly, please raise your hand and I can unmute you. Christine, you should be.
I think I unmuted myself. Yeah. Hi, my name is Christine. I work at Electrolabs, where we're building a catalog of tools for remote monitoring. So I'm curious about what played into your decision-making process to choose the tools that you did, which I believe were the Opal and the BioStamp by MC10. Just about your process for how you selected those two versus some of the other things that are out there.
Yeah, I can take that. So we looked at, and this is actually a core function of our group as well. We look at all the different devices that exist in the field right now. And one of the main things that drew us to the APDM device was solid battery life. Given, I think one consideration here was our study was in lab, so we didn't require an extensive period of time, but it was a device that's been used pretty well throughout a lot of the different research studies in the area, in the field, I should say. And secondly, on the BioStamp, I mean, I think the biggest attraction with the BioStamp is form factor. It's a flexible patch that you can put anywhere on the body. And so we were trying to assess also as well wearability aspects too. So that's why we looked at the BioStamp. Shama, you want to add anything as well?
Yeah. So hey, Christine, nice to virtually meet you. And so to add to what Nikhil said, there are a few aspects. I think one of the things that might, there's a figure in this paper that kind of shows all the device locations on the body. So this study was actually designed before Nikhil and I joined Pfizer. So kind of it was underway when we joined Pfizer. So we were not necessarily involved in a lot of the decision process, but we do know sort of what's sort of the thinking behind these choices. So as Nikhil said, APDM, one of the biggest factors for APDM was the fact that they have been widely used across a number of clinical studies. So in terms of just having the confidence that the system is accepted by the community. I think that was one reason. The other big reason from a technical point of view is that they have probably one of the best time synchronization when it comes to multiple devices on the body, and they maintain very high degree of synchronization throughout the monitoring period, which was in this case mostly in the lab setting, not at home. And I think BioStamp definitely with the form factor and sort of the comfort aspect, and I think that was so the idea was, I think, always, we want the system that gives us the best quality data in the lab for our experiments, and then we also want to keep an eye on a piece of technology that can be then used for the at-home kind of deployment part. So I think that was the rationale for choosing the BioStamp. And the BioStamp is also one of the few devices that you can literally place on any location on your body, and that was definitely one of the interests in terms of trying to capture as much data as we can in this study. So be a little bit more aggressive with device placement and capturing all aspects, and then figure out what is it that we really want to measure. And that's sort of what we try to do here in terms of reducing the scope of measurements.
Cool, thank you. Can I ask a follow-up question?
Please.
So one of the other tools you guys mentioned in the intro, or like the related work section, is the Kinsia from Great Lakes Technology. And I've seen they have a lot of published evidence, but I think one of their drawbacks is that their algorithm is proprietary, so you can't get access to that raw data. Do you have any comments on like whether that impacts whether people should choose that device? Is that a big downside of their product?
You want to take this one, Shama?
Yeah, so that's a good question. I mean, I think, at least in my mind, it's not the hurdle is not the fact that it's a proprietary algorithm. There's another device called Parkinson's kinetigraph, PKG for short, and they actually have a system that is very similar to what we kind of came up with in terms of a wrist-worn device that would be able to measure motor symptoms, tremor, bradykinesia. I think they also have some algorithms to detect dyskinesia. The algorithms are proprietary. I think one of the considerations for me, at least, is that if the system has gone through independent validation, so a lot of these systems are validated kind of just by the company through their own clinical studies or research, and I think there is an element of how much how unbiased was this work, right? So I think it's very helpful to have even if the system, the algorithms, the methods are not necessarily open source, as long as the system has gone through independent validation by other research labs that are not affiliated with the company. I think that adds a lot from the confidence point of view. Yeah, so I think that's.
I would add one thing as well to that, to what Shama said as well, and kind of taking it from the perspective from where we come from, which is big pharma in Pfizer. And I think one of the common threads that we've seen over the past 3 years that we've been there is that having other people validate the work, like Shama said, helps a lot with regards to getting approval from regulatory bodies like the FDA, which might be slightly different than the use case of a kind of consumer clinical care aspect.
Thank you. Super helpful.
Fantastic, Christine. Thanks for some terrific questions there. David, I see that you have a question. I've taken you off mute, I think, if you want to pose that to Shaimal and Nikhil.
Yeah, great. Guys, thank you so much. This is Dave Dansereau. Keep doing the great work you're doing in this area. My question is pretty specific to sensor setup. I read in the feedback from some patients, they certainly didn't like the sensor on their sternum, but related to the wrists and the feet, was the sensor on the wrist, was it at the wrist or was it on the back of the dorsum of the hand? I can't really tell from the figure.
It was, so the APDM sensor was worn like a wristwatch. So it was on the back of the hand.
Okay.
The MC10 device was on the inner side. So I don't know whatever you would call this here.
Yeah. Oh, okay. So, but it was, yeah. Okay. And then at the foot, where? Yeah,
so the APDM devices, the way they're worn, they're basically strapped to the shoe. So this device sits on top of your foot. It's essentially on top of the shoelaces. If you had shoes with laces, right? It's kind of, that's the position. And it's bilateral on both feet. We did not, for this work, we did not use any of the data coming from other locations. But yeah, so the setup, their standard setup is 6 devices, 2 on the feet, 2 on the wrist, 1 on the sternum, and 1 on the waist. So they use 6 devices.
Great, thank you.
There is a question from Joanna.
Yes.
Just want to read it out. So, are you planning to use the same device algorithm in future Pfizer studies, or are you considering other options? And if so, which ones? and what did the patients say about the device? Okay, so good question. So I, unfortunately, Pfizer does not work in neuroscience anymore. So we don't see in the near future us using this algorithm in Parkinson's. The interesting thing has been that this work actually helped us set the foundation for a lot of the work that came afterwards, not in neuro, but other areas. So things like, developing a modular framework, even some of the elements of this in terms of the algorithms that we implemented, like detecting hand movement, the machine learning approach for training a model to detect tremor. Those are all things that we were able to use and on a new data set, and essentially, sort of jumpstart our endpoint development process using these sort of standalone modules. So we are actually using those in current studies, and we will be using them in future studies as well. Regarding what patients said, we have actually a section, and that was one of the things that we cared about deeply in terms of understanding the patient perspective on using these devices. The way we designed the study, we mainly focused on the wearability aspects, and generally, we found that patients were, especially, I think PD patients are particularly interesting group. I think from this point of view, they're very passionate about kind of advancing the field and contributing to in whatever way they can, and are also at least the ones that I've encountered fairly interested in technology, and that might also be a factor of other factors like where the study was drawn, and recruitment, and maybe patients who participate in these studies are biased towards being interested in this. But the overall the response was very very positive. I think there was a slight preference for a adhesive like form factor, and I think one of the factors that definitely came through was that patients want devices that are monitoring them from a medical perspective or clinical perspective, to not be out there. I think there is a general preference that they should be discreet, and I think patches that are easy to kind of wear and blend in, I think are definitely kind of add their weight from that point of view. APDM, I don't know if you've seen the APDM device, but it's essentially like a black kind of box with a light blinking, right? So it doesn't look like a wristwatch. If you're wearing that around, you definitely would get questions about, hey, what is that? And I think those factors are important to consider, I think, because compliance is one of the biggest factors that will influence data quality in clinical trials. So yeah, that's my side. I don't know, Nikhil, if you have something else to add.
No, I think that was pretty spot on.
Fantastic. Appreciate that question, Joanna. Simal, or Simal, I apologize if I'm getting that wrong. You had a question in the Q&A, but Shimal, I think you just covered it. So Kathleen, I'm going to go ahead and unmute you so you can ask your question. You should be able to go ahead, Kathleen.
Maybe you could try doing it in the chat.
Yeah. Kathleen, I'm so sorry, we can't hear you. Are you able to type your question into the chat to make sure we get to it?
Oh.
Okay, we can see it.
So I'm wondering about passive measurement.
What in particular? I suspect you're probably typing. I'm sorry. Yeah, there you go.
Yeah, so that's an interesting question. So we actually explored a bunch of things in this in our work in PD. One was around kind of instrumenting these assessments. So can we instrument the UPDRS so that we are able to, instead of asking a human rater to rate give the scores, what if you had an algorithm that uses data from sensors, giving you a rating. So probably could be more accurate, could be, you know, more. So I think those are one of the, so we actually had an interaction with the FDA on this topic in terms of what is it that, you know, we brought to them some of the work that we have done in terms of data, developing, you know, the clinical studies that we run, and we asked them some questions about what would you find acceptable? Would you find these endpoints acceptable? And one of the feedbacks that I distinctly remember was that they would rather, I think it's more interesting, rather than just automating or, you know, making the existing assessments more accurate or reliable, it's more important to see functional changes. I think that the better use for technology is to see functional changes that are occurring at home and to the quality of life of these patients. So, you know, even going from, you know, here our work has been predominantly focused on assessing tremor and bradykinesia continuously. But I think the sense I got was that what would be more relevant actually would be to also connect the assessment of these symptoms with functional improvements. So because the tremor, you know, if their tremor constancy went down by a certain degree and, you know, they're less bradykinetic, how much does that impact in terms of what they're able to do or their quality of life? And I think that is honestly, personally, I find that as a much more powerful way of demonstrating efficacy of treatments than simply assessing, you know, ability to perform pronation supination, right, in the lab, whether based on clinical rating or using some instrumented approach.
Shaima, that's actually fascinating. I think we always think about sort of what is it, clinically meaningful change as being the yardstick, and really thinking about it being at the patient level and letting them tell us how the measure functions for them is terrific. And then I see that note from Kathleen. Good, I'm glad we got your question. I'm sorry about the mic. And then there's another question in the chat from Damien. I'll give voice to that, Damien. So his question is, why do you think there's no tremor during gait in your classification? And is that a technical limitation, or is that some kind of, you know, a medical hypothesis there?
Nikhil, do you want me to take this?
Sure. Yeah, great question, Damien.
Yeah, so that so there definitely could be tremor. I think we had a bunch of discussions with our clinical colleagues, you know, who have background in neurology, and the general consensus was that you know, it's so hand tremor can be present during gait periods as well, and that's absolutely true. But in terms of whether bucketing it as resting tremor versus action tremor, that was sort of a little bit of it wasn't clear where you would want to bucket it. And the second aspect in terms of, you know, keeping this sort of simple in terms of just focusing on periods when there is no hand movement occurring, allowed us to really hone in on the sort of the very pure resting tremor when they're, you know, I think it's very unlikely that you're engaged in situations where your hand is moving and you know, you would be considered at rest. So it definitely helped us, you know, twin down. And the other aspect that it helped from a technical point of view was in reducing false positives. So I think if you know, there's always a trade-off in these decisions. So whether you try to capture every aspect that you consider useful versus, you know, in this case, I would argue that 80 to 90% of the time that you would want to assess rest tremor would be in the situation where the individual is at rest, sitting, you know, in some posture, right? And so you could try to do really well at capturing that, or you could try to get that extra 10 or 20% of measurement in at the cost of potentially higher false positives. Because as soon as you try to say, okay, I want to also include, you know, periods like gait where you would have the hand is moving, but then there is tremor, you would potentially bring in periods where there could be, hand movement, they're engaged in some activity, and there is sort of, kinetic tremor that is kind of coming in, and you're bringing that into assessment. It would probably not be a big contribution to the overall assessment of resting tremor, but, I think that's just a design kind of choice.
Yeah, so I guess it would be a kind of a combination of both technical limitation a bit and also medical hypothesis a bit.
Fantastic.
So this is actually, sorry, just to add, this is actually really interesting in terms of, so we made these design choices, right? And if somebody else was developing these, the same exactly, they were targeting resting tremor and bradykinesia, you could make different design choices. I think one of the challenges in this field is going to be, is there a right or a wrong, or who sets these, is there somebody who needs to kind of set these conditions that you do need to measure resting tremor during gait, and that is important. And if an algorithm or a system is not providing that, then it is, that should be accounted for, right? And I don't have an answer to that. I mean, that's something that's worth the discussion in this community. Yeah,
I think that's a great point, Shamal. And I think, that's something that we definitely want to bring up as well with our interactions with the different regulatory bodies and get their point of view.
Shamal, that is indeed a great question. I wrote that one down, so it's very much on the record, and I'm glad you brought it up. We have time for one more question. I always try and finish a bit before the hour so folks can get ready for their next meeting, and it's from Supriya. The question, guys, is: have you considered comorbidities that could cause resting tremor? Moreover, have you considered revalidating your diagnosis with a secondary marker?
So I followed the first one. I'm a little confused about the second one. Let me tackle the first one. Nikhil, okay. Is it okay if I take a first stab? Sure. So we did not consider other comorbidities that could cause resting tremor, at least in this population. It's a good question. Actually, that's something that I'm not aware if there are other, in this population, if that is prevalent and is something that we should consider. But that's a good point. I definitely will have a discussion with some of the clinical folks that I work with and ask them that question.
I'm going to interrupt you for 1 second, Shamal. Supriya, if you're able to talk, I've set it up so you can unmute yourself if you want to sort of hop in and give some more color to your second question. Mm-hmm. I don't know if you're able to do that where you are. Let me give you a second.
Yeah. Thanks, Jen. Thanks, Shamal and Nikhil, for the wonderful talk. So my second question, where I'm really coming from, is there are vital signs that could point to Parkinson's, not independently as themselves, but which could be a sort of added evidence to the kind of biomarkers that you are looking for. So I'm just asking along those lines of, have you thought about other markers to kind of strengthen your diagnostic validation?
I think that's a great question, yeah. And maybe Shamal, I can take a first stab.
Yeah.
I think it kind of relates to the discussion that was a couple of minutes ago about quality of life and mobility, if I'm understanding it correctly. I think building up that evidence on other aspects of well that are more patient-centric, such as quality of life and mobility, might help in terms of being able to measure multiple other aspects of disease severity, along with the common ones such as tremor and bradykinesia.
Yeah, I think an interesting thing could be to look at relationship between these symptoms, these motor symptoms, and life space, for example, mm-hmm. and how that changes, if that changes. There are a lot of other factors that could come in as well. The other thing I wanted to just clarify is that our objective here was not diagnostic, and I think it's a distinction that we definitely like to make is at least in PD we don't believe that at least right now I haven't got any sort of feedback on whether this type of tools would be relevant from a diagnostic perspective. I think the study you would want to design would be very different, and so I think it's a question that at least was not on our minds in terms of this work.
I see. All right. Thank you.
Thank you.
Fantastic. Nikhil and Shaimal, you covered so much today, really gave some fantastic color to the amazing work you've published. Also, thank you for doing things like making that data available and for taking time with us today. All of the attendees who joined us, this was terrific. Such wonderful questions. I learned a lot. It's also just really nice. I know, or I suspect that almost everyone is dialing in from home. It was really nice to have a good opportunity to get this community together and sort of have some interaction during a tough time. I really appreciate all of you. Nikhil, I wonder if you could just advance quickly. Just a note for folks on the line. First of all, we will be making the recording of this available and the slides that Nikhil and Shaimal prepared, so you'll have access to those, including the resources they shared, which are fantastic. We actually, our webinar on inclusivity and innovation, and specifically really building diverse, thriving teams, was rescheduled to tomorrow. So if you are still looking for other opportunities to come and join us in conversation, please hop on the line tomorrow. Nikhil, if you could please advance again. I'm sorry to make you do this. Thank you. Our next journal club, hot on the heels of this terrific presentation, will be Brene Bent and Jessalyn Dunn, talking about their recent publication, also in MPJ Digital Medicine. They were looking at different sources of inaccuracy, specifically for wearable optical heart rate sensors. And then one final advancement for me, please, Nikhil. Thank you for your patience. The other opportunity is right at the end of the month, March 26th. We do have two of our members, Reggie Swift and Karen Fortuna, coming and joining us in a lightning speed crowdsourcing. These are 30-minute snippets. People come with questions and challenges, try and crowdsource from the wisdom of this amazing community. So that's what's coming up. Shaimal, Nikhil, thank you again today for just a tremendous presentation. Congratulations on a great body of work. Thank you everyone for dialing in, and everyone stay safe. It's great that we can still connect using these sorts of platforms, and I hope everyone is doing well. Thanks so much.
Thank you.
Thank you, everyone. Bye.
Bye-bye.